Hi, welcome to Biology classes with Lakshmi Chandra. As you know, we are dealing with transcription in eukaryotes. We have completed the initiation and elongation phase, and today I have come with the last phase of that is termination phase. So in order to get the previous videos related to molecular basis of inheritance and principles of inheritance and variation in this class 12 biology, please subscribe the channel, click on to the bell icon and press the all option so that you get all the videos of this particular topic. Now for more videos on the link, you can also click on to the description. Okay, in the description the links are listed, so you can just choose the one which you need. Let's focus on our topic, transcription in eukaryotes, the termination phase, that is the last phase. So just directly going into the termination phase, let me just continue it with the elongation phase. So let me just take you to where we have stopped in the elongation phase. So this is the area where we have stopped. This blue color that I have drawn here, this represents DNA, and this black color that is drawn, of course, with a tail. So you can understand that is our RNA polymerase, the key player of transcription. Now in order to stop this transcription, what we have to do is we have to remove this RNA polymerase from DNA. As long as RNA polymerase is there with the DNA, it will be moving along the DNA and it will be doing the process of transcription, elongation. So but then now we are dealing with how it is stopping, how this transcription is getting stopped all on a sudden. So for that, it's a bit complex process, but we'll learn that under 5 heads so that it will be easy for you to grasp, understand and recollect. Okay, so that termination we can see under 3 heads: dephosphorylation of Dsif, dephosphorylation of serine 5 residues, poly-A termination signal, cleavage of primary transcript, and last of course dissociation of RNA polymerase. So that means RNA polymerase will not dissociate all on a sudden. For this transcription to stop, there are other phases in between also. So you can see here, when you focus, it is written dephosphorylation. So in termination, dephosphorylation is the main thing, that is the main signal for termination. As you know, for elongation and for initiation it was phosphorylation. And phosphorylation of which residue or which part do you remember? Of course, it is the tail part. You call it a CTD, carboxy terminal domain of RNA polymerase. So this is the carboxy terminal domain. This tail part that I have drawn here, this is the carboxy terminal domain. And in this domain, there are some serine residues in the 2nd position, in the 5th position and all, about which I have in detail it was told in that initiation phase itself. The importance of this tail and the different residues there. So here we are concentrating on dephosphorylation of Dsif. About Dsif also it was told in detail in the previous video, in the elongation video. So that's why if you just subscribe it and if you go through the videos, it will be very easy for you to understand regarding these aspects clearly. So dephosphorylation of Dsif, how that is happening. So first step that we are going to learn in termination is dephosphorylation of Dsif, because Dsif got phosphorylated and because of which elongation happened. Now we are going to stop that, isn't it? So dephosphorylation has to occur for Dsif. So how this is doing, or who is doing this? This first one, 7-SK-SNRNP. This is a molecule which is carrying out the dephosphorylation of Dsif. So 7-SK-SNRNP. Ribonuclear phosphate. So 7-SK-SNRNP binds with Dsif. This is Dsif. So here 7-SK-SNRNP binds here. Because of this binding, a phosphate molecule which was attached to Dsif is now getting dephosphorylated. It is getting removed. And when this Dsif is getting dephosphorylated, at the same time what happens? PTEFB, I hope you remember this, PTEFB, the positive factor which is aiding in elongation, that also gets dissociated from the carboxy terminal domain. So the first step is dephosphorylation of Dsif, because of which a positive factor which was there in the tail part of the RNA polymerase, which was aiding in the elongation process, is now getting dissociated from the tail region. And instead of that positive factor, who comes and binds? Instead of this PTEFB, now in the dephosphorylated part here, NELF comes and binds. NELF. And NELF is doing just the opposite of PTEFB. It is a negative regulator of transcription. So this positive regulator, negative regulator about PTEFB and NELFB, everything was, NELF was was taught in detail in the previous session, in the elongation session. So if you want to go through that, please go through the session of transcription elongation in eukaryotes. Okay. So here the step is dephosphorylation of DSIFB by 7-SKSN-RNP. Because this phosphate is removed, the positive factor from the tail region is also removed. Instead, a negative factor comes and joins. So that is the first step. Now, because this negative factor comes and joins, another enzyme, serine 5 PPase, gets activated. This enzyme, what it does, it is doing the second step, that is dephosphorylation of serine 5 residue, as the name suggests, serine 5 PPase. It will dephosphorylate the serine 5 residue. Where is the serine 5 residue? Again, it is there in the tail region, that is the CTD, carboxy terminal domain of RNA polymerase, is having serine 5, is having the serine in the 5th position. So here again the serine in the 5th position also gets dephosphorylated because of the enzyme serine 5 PPase. Now when the serine 5 PPase gets dephosphorylated, what happens? Another set of proteins, CPSF, gets activated. CPSF stands for cleavage and polyadenylation specificity factor, CPSF. Now this CPSF will not get activated all on a sudden. It gets activated when a termination signal is reached. You know, initiation was there in the promoter region. For elongation there were signals. For elongation there were factors also. Similarly, for termination also there is a signal. What is that signal? As you know, in the DNA molecule, as the RNA polymerase is moving forward like this, new RNA strand is getting synthesized. In the 5' to 3' direction, new RNA is getting synthesized. As this RNA is getting synthesized, when an RNA molecule having the residue AA, UAA is getting formed. As this RNA is getting synthesized, when in that RNA a signal like AA, UAA is getting formed, that signal sends a signal for termination. So we can say that as a poly A, many adenine residues are there. So when many adenine residues are getting added up in the RNA which is newly formed, it is a signal that this termination or this transcription is going to end. That is a signal for this RNA polymerase to state that yes, now you have to leave from this DNA. Now this process is going to end. So that's a signal. So what is that signal? Poly A signal. Where is that formed? It is formed in the growing RNA chain. So as this signal 5' AA UAA 3', this termination signal is reached, to that termination signal immediately the CPSF binds. Cleavage and polyadenylation specificity factor binds to this particular signal. What is that signal? Termination signal. CPSF binds to it. Now, as soon as CPSF binds to it, so I can just put here, this is the growing RNA, this is the 5' end, it is growing, and this is the signal, termination signal. To this signal, who comes and binds? CPSF. Cleavage and polyadenylation specificity factor comes and binds to this particular signal. And in turn, the CPSF recruits another factor, CSTF. CSTF stands for cleavage stimulating factor. Okay, the CPSF recruits CSTF. CSTF. Now this CSTF binds over another signal called as U or GU signal, which is there downstream towards this particular termination signal. Okay, so these are the two things. CPSF binds over termination signal and CSTF binds over another signal that is further downstream towards the actual termination signal. Now the binding of CPSF and CSTF is very crucial because that decides the point where the primary transcript has to be cut from the growing chain and it has to be detached. So actually the cutting takes place here. This is the area where the cutting takes place. Or instead of cutting, we can use the word cleavage takes place. That is why the word here, cleavage and polyadenylation specificity factor or cleavage stimulating factor. So this is the importance of this. Cleavage means cutting. Okay. So because these two comes and binds here, there occurs cleavage in between these two. So when this is cleaved, what happens is, in this diagram, if I am showing you, it is cleaved here. On the upstream, on the downstream, what is there? U signal or GU signal is there. So between these two, there is a cut. That cut is what is shown here. And since it is cut here, this growing RNA chain will be detached from this DNA and it will come out. Okay, since it is cut over here, it comes out. So now this growing RNA chain, I'll just show it here. This growing RNA chain is now like this. 5' end. Then this growing chain. And here you have the termination signals AA, UAA. And then afterwards there is a cut. So till here we got. Okay, we got till here. Now immediately after this, what happens? Immediately after this, that is, this end is the 3' end. Okay, this end is the 3' end, this is the 5' end. Immediately after this, what happens is polyadenylation. That is, many adenine residues are getting added up. Many, maybe 200s or so adenine residues are getting added up. And this is done by an enzyme called as A Polymerase. Very true to its name, isn't it? A. Many adenine residues are getting added up. A Polymerase, the enzyme, keeps on adding many adenine residues towards the 3' end. Of course, this is a modification also of this primary transcript. Now this RNA that we got here, we cannot call this as mRNA, we call it as the primary transcript. Primary transcript. It has to undergo some more modifications to make it the fully fledged functional mRNA. Now you cannot call it as mRNA, you can call it as a primary transcript, or it is also called as heterogeneous RNA, or in short we call it as hnRNA. So what we got immediately after transcription, it is not the mRNA, but it is primary transcript or hnRNA. Why it is called as primary transcript? Because it is not functional yet. Okay, it didn't start functioning. And why it is called as hnRNA or heterogeneous RNA? Because this primary transcript that we got is having regions which are functional. It is having some other regions which are non-functional. So two regions it is having, functional regions and non-functional regions. So the areas which I have drawn with blocks like this, these are functional regions, or we call them as the exons. Functional regions are called as exons. And in between there are regions which are non-functional. They are called as introns. Again you can see here exon is there, intron. Again exon, intron. So non-functional regions are called as introns. Introns or intervening or interrupting sequences. So because this primary transcript is having functional region and non-functional region, you call this as the hnRNA. So if you want to get the correct mRNA from the hnRNA, what we have to do is we have to remove all the introns. All the introns one after the other and add up the exons, combine the exons. So removal of introns and combining of the exons is called as splicing. Splicing. This is a modification that is done in hnRNA or the primary transcript. That is removal of all the non-functional regions or introns and then thereafter adding up all the exons. That is called as splicing, and it is done by an enzyme called as spliceosome. This also in detail will be dealt with in another video regarding post-transcriptional modifications. That is, after transcription, what happens to this hnRNA or mRNA, that will be dealt with in another session as post-transcriptional modifications. So for now you have to understand the modification is many adenine residues are added by the enzyme A Polymerase, and this speciality of this enzyme is that this enzyme does not require any template. You know, usually the polymerases, suppose it is DNA polymerase, it requires a template for it to add up the nitrogenous bases. If it is RNA polymerase also requires a template to add up the nitrogenous bases. But the speciality of A Polymerase is that it doesn't need any template. It keeps on adding adenine residues. At the end of immediately after cleavage, it keeps on adding adenine residues. So now you got the hnRNA cleaved from this DNA. It has come out as a primary transcript with exon, intron and a tail of adenine. This you call it as a tail of adenine, or you call it as a poly-A tail. Poly-A tail. That is there in the tail part or in the 3' end. In the 5' end, there is a cap. So we can say the primary transcript is coming out with a cap and a tail. And what is this cap and tail for? It is for protection. I told you regarding the tail. What is the tail? Poly-A tail. That is for protection. And again a cap it comes with. And this cap is formed as soon as it begins the process of transcription. This cap, the name of the cap is 7-methyl guanosine cap. 7-methyl guanosine cap, or simply we can say methyl guanosine cap. Okay, so there's a cap, tail I told you, and then removal of introns and joining of exons. Regarding the detail of what is this cap, how it is getting added, why it is getting added, and then regarding the importance of this tail, and then how spliceosome is doing the function of splicing, everything will be dealt with in the next session. So in this session, now I have to deal with this particular part, dissociation of RNA polymerase. So far I have told you our primary transcript has come out. Okay, fine, we got the primary transcript. Now we need modifications. But the story is not ended here. The story continues. Why? Because our RNA polymerase is still attached to DNA. As I told you in the beginning, as long as RNA polymerase is attached to DNA, it will keep on doing transcription. Okay, so the last final step is dissociation of RNA polymerase. So this dissociation of RNA polymerase is well explained with the help of Torpedo model of termination. There are many theories for dissociation of RNA polymerase, how it is getting dissociated, but this particular model well explains it and it is widely accepted too. So let's focus on this one. Torpedo model of dissociation of RNA polymerase or Torpedo model of termination of transcription. So here the key player is XRN2 enzyme. XRN2 enzyme. It represents exoribonuclease 2 enzyme. This is the key player. So we have learned, we have this DNA transcription bubble is what is drawn like this, and onto this DNA template we have the RNA polymerase placed. RNA polymerase is here, placed over the transcription bubble like this. RNA polymerase is having a carboxy terminal domain like a tail. Okay, now onto this all the dissociation factors are present. Now new RNA molecule is getting formed in the 5' to 3' direction, and as soon as the AAUAA signal is reached, this RNA is getting cleaved. Now this RNA is no more present here, it is getting cleaved by the CPSF factors, CSTF factors and all. Now this RNA was having a cap in the beginning, 5' end, and it is having exon and introns here, exon and introns, and then at the end it is having termination signal followed by poly A tail. Okay, but still who is here? RNA polymerase is here, and as the RNA polymerase is moving forward, it will produce another set of mRNA also, having again a 5' end and a 3' end. It will produce another set of mRNA. But the basic difference between the mRNA produced here first and the second one is that the second mRNA in the 5' end there is no cap. There is no methyl guanosine cap. So methyl guanosine cap is found only in the first mRNA or first primary transcript that is formed. It is lacking in the next mRNA or next RNA which is getting formed. The RNA polymerase is moving forward and it is producing new RNA molecules, but the second one which is getting formed, that is the one which is formed after cleavage, will not have a cap. And if an RNA molecule is formed without a cap, what happens? Without that cap, without that protection, what happens? Immediately, the exoribonuclease 2 enzyme, it recruits the factors called as P54NRB PSF complex. This is a complex, this is an enzyme complex recruited by XRN2 enzyme. So this particular complex comes, and what it does is it will find out the uncapped RNA transcript. The one which is not having that cap, the one which is coming without protection. It finds it out, the uncapped RNA transcript. And what it does? It starts chopping, keeps on cutting, chopping that RNA which is not having this cap in the 5' to 3' direction. So this is the RNA that is formed in the 5' to 3' direction without cap. So this particular enzyme XNRN2 recruits that particular factor, and because of which it chops the RNA which is getting formed immediately. And this chopping is very swift, very fast, faster than the rate in which the RNA is moving forward. The chopping will be faster than the rate in which the RNA is moving forward. So As the RNA is getting produced, it is getting chopped, and again it is produced, it is getting chopped. The rate of chopping is faster. See, chops RNA transcript swiftly in the 5' to 3' direction. And because of this swift chopping, as the RNA is getting formed, it is getting chopped down. What happens is the RNA polymerase, which is moving forward, undergoes a conformational change, undergoes a change in its shape. It recognizes, oh, whatever RNA I am producing, another enzyme is coming and chopping it out. Why should I produce it? Why should I stay with this DNA longer? I will not stay with this DNA. I'll better move out. So actually, because of this swift chopping, that is the torpedo model, okay? Because of the swift chopping of the uncapped RNA transcript, which is formed subsequently, this RNA polymerase undergoes a conformational change, and because of that, this RNA polymerase is getting pushed out or pushed away from the DNA. And as the RNA polymerase is pushed away from the DNA again becomes double-stranded, and the process of transcription ends there. So I hope you have understood the process of termination in eukaryotes. We have been learning this quite a long period of time. Transcription in prokaryotes, transcription in eukaryotes, initiation, elongation, termination phases. Termination phase, I have told you, it's very simple. It's the basic mechanism you have to remember is dephosphorylation. Dephosphorylation of Dsif. Because of that, instead of a positive factor, a negative factor comes and binds. When that comes and binds, the serine is getting dephosphorylated. When the serine is getting dephosphorylated, the cleavage factors, 2 of them come and bind. And as they come and bind, where do they come and bind? They bind at the termination signal, the AAUAA or U or GU signal, they come and bind. When they come and bind, there happens a cleavage. After the cleavage, you get the primary transcript. And in the primary transcript, in the tail end, poly A, many adenine residues are getting added. And the importance of 5' capping, poly A tail, removal of introns and exons, and exactly what is this functional and non-functional, what do you mean by that, and all will be dealt with in the next session. So immediately after this is getting formed, RNA polymerase is still transcribing. It is still doing the process of transcription, but we don't need it, isn't it? But what is the mistake it is doing? It is producing the RNA without a cap, without a protection. Immediately it is recognized by XRN2. It is chopping it down, chopping it very swiftly. So the RNA polymerase is no more feeling like to be with the DNA. It undergoes a conformational change, it comes out, and the process of transcription stops there. I hope you have understood this very well. See children, always understand this as if we are going through a journey. Initiation, in the initial phase when a child, when the child is born, okay, the child is learning to crawl first, and then to stand up, and then to walk and to run and all. At that time, so many people will be there surrounding him or her to guide him or her. So that's what we have learned. There are so many initiation factors. And then as the life proceeds, there will be our parents and teachers and all to guide us. So that is how elongation factors are there. Suddenly we have to do things on our own. So sometimes we make mistakes, we may just come out of that and we may continue our life. But for everything there is a end, there is a termination. Similarly, for our transcription process also, there is an end. But here, actually this has not ended. I'll come with another session telling you how this modifications, this cap, this tail, intron, exon, everything is getting processed. So wait till then. Thank you all.